Let's look at the steps of mitosis. We're going to start with a cell in the G2 phase, or the late interphase stage. We look at the chromosomes in the nucleus and we see they are not fully condensed. We see that there are two centrosomes because these will form the spindles in order to separate the DNA. Before we begin, the cell is going to check to make sure that the DNA has been completely replicated. The first stage of mitosis is known as prophase. Here, the chromosomes condense and become visible. And so you can see here that we have a chromosome with two sister chromatids and another chromosome with two sister chromatids. In this case, the red chromosome is from the mother side, the blue chromosome is from the father side. We have another chromosome with sister chromatids and another chromosome with sister chromatids. We see that the spindle forms as the centromeres move to opposite poles. And that's this part right here. There's the centromeres. They're moving, and here are the spindles forming. The nucleus begins to break down. And so here you see the dotted line representing the nuclear envelope is breaking down. Before we continue, let's explain exactly what we're doing here. We're going to look at a human karyotype before synthesis phase. A karyotype is simply where we're showing the chromosomes in the human. Humans have 23 chromosomes. Here is chromosome 1. These are numbered based on their size. Notice that you have a version of chromosome 1 from the mother side, and you also have a version of chromosome 1 from the father side. And so for this lecture, we're showing the mother's chromosome in red and the father's in blue. You also have chromosome 2, which is slightly smaller, chromosome 3, which is smaller, 4, which is smaller, and so on and so forth. And we go all the way to these smaller chromosomes down here until we hit chromosome 22. What about the 23rd chromosome? These are going to be the X and the Y chromosomes. So notice if you have two X chromosomes, you are female. And if you have an X chromosome and a Y chromosome, you are male. We want to see here that this human karyotype is before synthesis phase, so that these chromosomes only have that single chromatid, and you do not see the double bars. After synthesis phase, the karyotype will change and will look different in the sense that the DNA has been replicated. So here we have an example of a male karyotype after S phase, after synthesis. And so let's look at chromosome 1. This one right here is chromosome 1 from mom, but notice there are two sister chromatids now. The information has been replicated. There is twice as much DNA there. Here is the chromosome 1 from dad. Again, you see the double bars. Same thing with chromosome 2, and all the way to the X and the Y. That's what synthesis or S phase is all about. And this is just a reminder down here that after S phase, you would have the sister chromatids, which are identical to each other, and they are being held together by these kinetochore proteins. So the human karyotype has 23 pairs of chromosomes. However, for the sake of simplifying pictures and not drawing too much, we follow mitosis for 2 of these 23 pairs. In other words, we are looking at chromosome 1 from the mother, which is this larger red one, chromosome 1 from the father, which is this larger blue one, and these smaller chromosomes, you can take your pick as to which one you want to imagine. So this one right here could be, let's say, chromosome 10 from the mother, and this one right here would be chromosome 10 from the father. And so just to realize that that is what we're looking at inside this nucleus where the envelope is breaking down during prophase, where these spindles are reaching out, getting ready to orchestrate this cell division. And so this is what we left off at prophase. This was the picture we finished with. The second phase of mitosis is known as prometaphase. Here we see that the nuclear envelope is broken up completely, which means that the chromosomes are now free to scatter around the cell. They are no longer confined to the nucleus. The spindle fibers attach to the kinetochores on the chromosomes. And so here you see that the spindles are reaching out, grabbing the kinetochores. The third phase in mitosis is known as metaphase, and this is where the spindles will align the chromosomes along the equator of the cell. So the equator of the cell is in the center, meta means middle, and you can see that the chromosomes are aligned down the middle. In the fourth phase, known as anaphase, the microtubules from the spindles start to retract and they pull the sister chromatids apart. The centromeres split as the sister chromatids separate and move to opposite poles of the cell. You can see that these sister chromatids are moving to one pole, these sister chromatids are moving to the opposite pole. In the fifth and final phase of mitosis, known as telophase, telo means end of, the nuclear envelope reforms at each pole. This dotted line is representing the nuclear envelope on one pole. This dotted line is representing the nuclear envelope at the opposite pole. The chromosomes begin to decondense and the spindle apparatus disappears. You can see it here that it is shriveling up as it is retracting itself. What is interesting about this final step is that this is still one cell, and you can see it by this boundary, but this single cell is developing 2 nuclei simultaneously. After telophase, strictly speaking, we are at the end of mitosis. So telophase is the last step of mitosis, which means that cytokinesis, strictly speaking, is after mitosis. Cytokinesis is the division of the cytoplasm into 2 cells. So cyto meaning cytoplasm, and kinesis meaning movement or separation of. What you can see here is that we form this contractile ring, and this contractile ring is the boundary of these 2 cells that develop. The nuclear envelope here is fully formed. The chromosomes are decondensing, the microtubules are retracted. This contractile ring will cause the separation of these two cells entirely. So here we have one daughter cell, and here we have the second daughter cell. These two cells are both now in their respective G1 phases. At this point, the cell cycle begins anew. We are now back to this point right here, where these two cells will begin their respective G1 phases.